It is a pleasure to be able to present the findings, the objectives and the findings of the ASSURE study this morning. This slide summarises my disclosures. There continues to be immense interest in the development of therapies that target HDL. And for many decades, so many of us in the lipid field have wondered if the simplest way to approach an HDL therapeutic might be simply to upregulate the synthesis by the liver of its major protein ApoA1. And in theory, increased production of that protein would generate new HDL particles, go out into the bloodstream, promote cholesterol efflux, reverse cholesterol transport, and in theory, reduce the size of plaques and ultimately reduce cardiovascular outcomes. And so we now have the first oral agent that is proposed to be able to induce hepatic synthesis of ApoA1. We took it into a large intravascular ultrasound study to understand whether it did regress atherosclerotic plaques. So what we present here today is 323 patients with symptomatic coronary disease with low HDL cholesterol levels who were randomised in a 3-to-1 fashion to treatment with the ApoA1 inducer RVX 208 100 milligrams twice daily or placebo. They were treated for 26 weeks. So in the context of an oral intravascular ultrasound study, this was quite a short study. As you can see, they were seen quite frequently. They were seen 2 weekly for the first 8 weeks and then 3 weekly for the remainder of the study. So that equated to a large number of visits during the course of the study with intravascular ultrasound performed in the same coronary artery at baseline and at the end of the study. This slide summarises the biochemical effects. In the RVX group, we saw significant increases in ApoA1 and HDL cholesterol. We saw a significant reduction in LDL cholesterol by about 16%, but as you'll see on this slide, we saw exactly the same biochemical effects in the placebo group, which was quite striking in the course of a 6-month treatment period. This slide summarises the primary efficacy parameter, which was the change in percent atheroma volume compared with baseline in the RVX group. We saw a reduction in that parameter by 0.40. It just failed to meet statistical significance of a p-value of 0.08, but as you'll see from this slide, essentially the placebo group did the same thing with a reduction by 0.30 when the p-value between the groups was 0.81. We saw a similar message when we looked at the secondary endpoints, atheroma volume throughout the whole segment of the vessel studied, and just in the 10 millimetre segment that contained the most amount of plaque. Again, numerically greater regression with RVX 208, but essentially no statistically significant difference from placebo whatsoever. This slide summarises the clinical and adverse biochemical effects. We did see numerically less cardiovascular events in the RVX group, although that did not meet statistical significance. This was not powered to look at clinical events. Most importantly, and consistent with prior observations with this compound, we saw an increase in liver transaminases in most patients treated with RVX 208, and 7% of patients in that group, in fact, had a ALT or AST greater than 3 times the upper limit of normal. So, in conclusion, we saw increases in HDL cholesterol and ApoA1 and a decrease in LDL cholesterol compared with baseline with the drug, but that did not differ from the placebo group. For the primary endpoint, we did observe a trend to regression with the drug. Again, no statistical significance compared to placebo. Similar finding for the secondary endpoints, and we did observe, as we saw with prior studies, that RVX 208 administration was associated with liver enzyme elevations as previously described. And I think there would probably be 3 take-home messages from our perspective to this study. The first is that this drug had been developed with the thought that it would be a potent oral compound from the perspective of an HDL therapeutic and would be comparable to the infusional approaches that we'd seen previously regress plaque. That's clearly not the case. We saw no biochemical or plaque increment whatsoever above and beyond the placebo group. So it does not appear to be a potent approach to HDL therapeutics. The second is the placebo group here did extraordinarily well. Great biochemical changes, a very good trend towards regression even within that group within 26 weeks. And I think it highlights the importance of treating patients well. These patients were seen extraordinarily frequently during the study. And so it really does tell us that we are getting greater and greater benefits with greater use of established therapies. And perhaps the third endpoint would be that we've done many of these studies over a much larger, longer time frame, 24 months. What we observe here is that, particularly if you look at the placebo group, there really is that trend towards regression at 6 months. It's not to the extent that we see at 24 months, and maybe what it tells us is that all that regression that we saw in those longer studies of high-dose statins, it wasn't all front-loaded. That regression really occurs over time, and it really continues to highlight the importance of compliance with established medical therapies in our patients with coronary disease. Thank you.
Thank you very much. Will you pose your questions for Dr. Nichols?
Bruce Jansen from Cardiology News. I understand your commitment to this HDL hypothesis, but I don't see it. There was the huge NIH niacin study that was negative, so I'm going to ask a neutral party, Dr. Batt. Dr. Batt, is the HDL hypothesis dead? It seems like all the news is negative.
I think that's a good question. The hypothesis has always been a good one, but I believe in patients that are statin treated, no one has really shown an incremental benefit of HDL raising. Of course, there's older data in patients not on statins that drugs like niacin are useful, but they also do have an effect on other lipid parameters like LDL. It's interesting, the Mendelian randomization work that's been done for the most part, and that's just a way of looking at genetics and seeing how high people's HDL levels are at birth and their lifetime exposure, for the most part have not shown an association between HDL and cardiovascular outcomes. So it does appear that low HDL is a risk marker, but it may not be a modifiable risk factor. So I think for the time being, it remains a hypothesis with a lot of holes in it. And I think moving forward, probably we'll have more to gain by incremental LDL reduction, even beyond the levels attained with statins, than with just trying to raise HDL, unless those HDL raising compounds we study in the future also have LDL reduction or other properties.
Would it be fair for me to perhaps
like-
make one comment as well?
You can respond also.
Yeah, I think the answer really is we haven't really tested the HDL hypothesis, and that becomes the single biggest challenge in these trials. That the only studies that have truly tested the HDL hypothesis were the studies where you directly infused HDL. You didn't do anything else. And in those studies, you saw rapid regression of disease. Now, we've seen none of those programs advance to the next step, which is the clinical outcome trial, which will be the ultimate validation of that story. But for every other clinical trial failure, whether it was torcetrapib, whether it was 2 niacin studies, whether it was this study, you can make the solid argument that there were many other things going on in those studies. We saw no incremental benefit on any HDL parameter in this study whatsoever compared to the placebo group. So one could make the assessment looking at those slides, have we really tested the HDL hypothesis with this drug? It did nothing incremental whatsoever to HDL. So I agree. I think this is a field that continues to have a lot of uncertainty placed by large trials that haven't worked out the way that we want them to. But we continue to think that the HDL hypothesis needs to be tested, and I think ideally the way that will ultimately happen is by infusing those drugs. I do agree with Deepak completely that the consistency across the board has been that LDL's bad, and if you lower it, it's a good thing. And I think that we've got a lot of studies going on in the PCSK9 inhibitor field. We'll see if they're beneficial by pushing the boundaries. We've got 2 potent CETP inhibitors that are moving forward that have dramatic effects on HDL cholesterol, but they lower LDL as well. So it may be that they work just simply because they do that. And the final comment I would make is that the genetics for HDL cholesterol is negative. The genetics for HDL may be a very different thing, and the HDL cholesterol only tells us one part of the story about HDL. Larry?
Isn't it possible that you're testing a hypothesis that you don't really understand at this point, that Dr. Bott talked about the correlation of HDL, but no one really understands yet how it works. So how is it possible to know how to manipulate it to get an effect?
Well, I think there's 2 points embedded within that. The first point is that it may not be as simple as LDL. In general, LDL is fairly straightforward, that it's bad and whatever you do to it tends to have a good effect. And it may be that because the functionality of HDL is more important than the HDL cholesterol, it may be that the type of HDL and therefore the strategy by which you modify it may be more important. So you're absolutely right. We may have just tested the wrong approaches to date, which is why I think that the field is not dead from that perspective. The other element becomes how are we evaluating these compounds. We need to get something to a phase 3 trial, obviously, to reduce cardiovascular morbidity and mortality. There's been a proliferation of different techniques to look at different functions of HDL. but we don't know which of them actually benefit human beings in vivo. And so that's why our interest has always been that the final common pathway of all of those functions, whether it's cholesterol mobilization, reduction in inflammation, whatever you want to call it, has to be to favourably modify plaque. And we've known in 3 studies that if you infuse HDL, it regresses plaque. And so it seems to me that an HDL-targeted therapy, to be able to work in a phase 3 trial, should regress plaque along the way. It was a pretty good story here, and it didn't work. And I think that will be a similar pathway to try and develop an HDL therapeutic moving forward.
Yeah, because indeed the drug didn't induce synthesis of ApoE1.
So there's preclinical data, obviously, in cells, and there's a big difference between going from a cell or an animal to a human being. And we saw in early studies, we did see some increases compared to placebo in ApoE1 and HDL, and large HDL in particular, suggesting that there was cholesterol mobilization. But we saw nothing here. So I think that we have to call this a neutral study, and we need to say that we haven't tested the HDL hypothesis with this compound in this study.
Okay. You want to ask?
So this question of HDL functionality for future studies, how are you changing your direction for the future?
Well, I think that there will be a lot of interest in cellular techniques early on in studies, but we need to be able to move compounds that make sense, that appear to be safe, and that appear to have beneficial effects on a number of functional assessments in cells and animals and whatever. They need to get to early phase 2 studies where we can look at imaging, because HDL and LDL are 2 fields where favourable effects with those drugs should translate to favourable effects on atherosclerotic plaque.
Okay, thank you very much, Dr. Nichols.
Thank you.